Hi everyone, this is Michelle from Methylation Madness, co-author, and I am sitting here today with Dr. Terence Dulin. And I decided that for this video, I was going to take a couple of questions from Facebook and see if Dr. Dulin could give a little bit more clarity to some of the questions, some of the comments. So, hi Doc.
Hello, Miss Michelle.
How are you?
Very good today.
Good.
Before we get started, there's some things I need to kind of clarify.
Okay.
I'm looking at a lot of the questions and I'm looking at a lot of the things that they're basically talking about. It comes to mind that a lot of people just really don't understand what the science of genetics and things are actually doing. They don't really understand what they're reading in all of the SNPs and everything that they're being put out there. When you look at the research for SNPs, okay, which are genetic variants or non-genetic variants, depending on their classification, you run into a single problem. Most of the research that's done has to be double-blind and carefully peer-reviewed, which means there's very little evidence beyond what that one paper tells you. Now, that doesn't mean that's the entire story. It means it's one paper, one person's opinion on that particular gene. It's not written in stone, and it's basically going to change as more papers and more things are published. So this is right off, people have to understand what they're reading has to be taken with a grain of salt because it's not the whole answer. Secondly, I hear a lot of talk about the A1298C and the C677T, and everybody is all talking about these as the main thing. You have to understand that there are many more MTHFR genes. This is not the exclusive piece that these two are the whole story. They're not. There are many other ones that do different things, but they haven't been researched, which is why they're not prevalent in the literature or the research at this point. So once you start to understand that, you get an idea that everything you're reading has applicable pieces, but it's not the entire story, and you cannot do a whole treatment protocol on one gene. That is impossible and crazy to even attempt. Okay, just because you have a 1298C, you may not have any single problems at all, because you may have a BHMT gene that picks up the slack and takes care of that, and therefore the MTHFR is not that prevalent or that important at that point. Same thing with the 677T. You may find that one is also in there and you get no symptomatology whatsoever, which drives a lot of the researchers crazy. You may have these genes and yet your homocysteine is completely fine. So why is that? Because the genes are only part of the whole story. Human beings are not a single gene, a single piece. This comes out of the medical model of the way that medicine works. Medicine works on the basis of identifying one mechanism, one problem, one chemical piece, and that's it. But they treat on the basis of a medication that affects that specific mechanism, which is why they boil it down to one piece. Amino acids cannot be used in that way. An amino acid may have thousands of different uses in the human body, in which case putting it in to try and get one specific mechanism to change requires other things to be brought with it as a group. If you don't bring it as a group, you're basically just saying, here, I'm going to throw some confetti out the window and I hope it gets on the car down there. Okay? It's not going to basically happen. And a lot of these problems people are having because they don't understand what hyper and hypomethylation are. Hyper and hypomethylation really don't relate to MTHFR. What hypermethylation is an excessive utilization of methyl groups in your system. Methyl groups are created basically by changing a carbon atom with 3 hydrogens on it from one molecule to another molecule. In doing so, you change the second molecule to something entirely different, so that that second molecule basically can be predominantly either detoxed or it can do a separate different function from the original molecule. That's what methylation is. Now, methylation gets involved in a lot of other enzymes, and we're going to discuss some of those today. The COMT is one, okay, that's very prevalent. It has a lot to do with detox, but it also has a lot to do with control of breakdown of neurotransmitters. Then you have other genes that are methylating. Just off the top of my head, you have the PUMT, you have the HNMT. These guys are different because the PUMT is responsible for changing noradrenaline into adrenaline, which if you're hypermethylating, that gene becomes important because you could spike adrenaline and that can affect everything from blood pressure to tachycardias and heart rates. So you have to be a little cautious as to what you're looking at there. HNMT is one of the major sources of the removal of histamines from your system. So it has a direct effect on allergic response, allergic activity, and anaphylaxis. A lot of you talk about hypersensitive allergies. If you are low methylating, then the HMT gene is not going to be activated. Histamine methyltransferase enzyme that it controls is not going to break down histamine correctly, and you're going to have a higher circulating level. Combining that with a high dietary source, you're now going to be primed for lots of allergic sensitivities and things. So right off the bat, people have to start understanding that this is not a very simple one-size-fits-all, one thing does this and that's it. It doesn't. And it reacts differently in each person. Each person is a unique little biochemical engine. We have our own biochemical pattern that is related directly to how our specific metabolism works. Think of it, a Volkswagen and a Ferrari are a car. They're both cars. But the same oil that you put in a Ferrari, you wouldn't want to put in a Volkswagen. And the same gas you put in a Volkswagen, you wouldn't want to put in a Ferrari because it's not going to run correctly. So this is how you have to start thinking about human beings. Individual mechanisms doing individual things. I don't write a lot of papers and things on that basis. If you were to try and put together any two people that have the exact same chemistry, after my 50, 60,000 people I've seen, I've never seen two exactly the same.
Even twins?
No, twins is the same thing. They have a different physiology, which is why you'll always hear the thought one twin is a little dominant, the other twin is not. That's because their physiologies are different. And when you look at different sets, you get different understandings that their physiology is just this bit or that bit. They may look the same, but they do not function the same.
So there was a question on Facebook, and it was about mood, 5-HTP, TMG, and SAMe. What do you have to say about?
Now you're talking about a massive amount of chemistry that you're affecting in one shot. TMG, otherwise known as trimethylglycine, is a betaine derivative. Okay, it's basically betaine. What it does is it takes homocysteine and it recycles it back up to methionine via the BHMT pathway, which is the BHMT gene. TMG, when it makes more methionine then picks up, ATP through the MAT1A, and it becomes basically SAMe. So if you're putting in TMG and SAMe, you're really cranking up your methylation system like crazy. That's good and it's bad. And why is it good and bad? Well, if you crank up the methylation system very high, you're going to get effects on all of those genes and enzymes I was talking about before. Specifically, you'll get a lot of effects on COMT. Okay? Now, COMT is a whole separate conversation, which we'll have in a minute. But COMT is responsible for doing many things. It breaks down neurotransmitters. Dopamine, norepinephrine, epinephrine, serotonin, histamine are all broken down by COMT, as is testosterone, as is estrogen. So, cranking up your methylation chemistry will have a direct effect on hormones and neurotransmitters. If you're not resupplying the neurotransmitters, you will burn them out after a time, which is why a lot of doctors are running into the problem anywhere from 3 weeks to 2.5, 3 months. Okay, all of a sudden the systems seem to go a little crazy and fall apart.
I've seen that on Facebook, where people are taking methyl something and then they feel great, and then all of a sudden they'll say, I've been taking it for a few weeks and now I feel awful.
Right, see, this is the boost in adrenaline that always happens from the PUMT enzyme. You get a spike in adrenaline, everybody feels on top of the world. Yes, I'm on speed, I feel great, okay? However, if you're not refilling the top of the pathway, then the pathway burns out, and now you're going to get hit with more anxiety and depression. You're going to have vision problems. You're not going to be able to focus and think correctly, and you're going to suddenly think you developed ADD.
While you're talking about symptoms, Doc, somebody spoke about...
Wait, we're not done with this one.
Sorry.
You're trying to rush, and you're not even there yet. So TMG, right off the bat, does not remove homocysteine. What it does is it recycles it. So if you keep recycling homocysteine, everybody goes, great, I couldn't get the homocysteine to make methionine. Don't forget there's a dietary component. Right. Methionine comes from food. Right. So if you're eating a high diet that's loaded with fish, you're going to have a lot of methionine coming into your system. The more methionine comes into your system, the more ATP is going to be required to activate it. It's going to make you tired if you don't make enough ATP. It's also going to continue to burn down your neurotransmitters. Now, does this happen in everybody? No. This only happens in the people who have these defects in the system. COMT is a whole nother ballpark in the way of genes. It has tremendous amounts of functions, and there are many, many, many of them. It's not just one or two of them, and they all have slightly different areas of the body, slightly different chemistries that they affect directly. That's what makes COMT interesting. Now, there's variants that lower CMT's function and drop it all the way down to 25%. At that point, that means over-methylating is basically not going to do very much to that enzyme, and you're going to overload things in your system really fast. Okay? You're going to jam up those enzymes until they don't function anymore. That's not going to be beneficial in any way, shape, or form. COMT is also responsible for liver detox. There are many different pathways of detoxification in the liver. Okay? The numbers of things in the liver, basically, you have NAT, you have SOD, you have all these other pathways to detox different types of compounds. And that's not even considering the cytochrome P450s. Now, the research on cytochrome P450s right now is being done strictly for pharmaceuticals because pharmaceuticals are what metabolize through the cytochrome P450 enzymes. This would be the ones that people would recognize as CYP1A1, CYP1A2. Oh, it has to do with coffee and estrogen. Yes, as far as the research knows, but these were put into our body long before people were drinking coffee and basically back in the day. So they're more related to plant compounds, which gives us the ability to have a diverse diet. These break down different substances found in herbs and plants. Herbs and plants are basically loaded with chemicals. Plethoras of different types of metabolisms go on from eating them. These cytochrome P450s and the detox mechanism prevent us from dying from eating a piece of fruit, okay, or eating a plant and dropping dead because it has something in it. Okay, just because it's natural doesn't mean it's safe. Belladonna is really natural and it'll kill you pretty fast if you eat a lot of it. Okay, cocaine comes from the coca leaf. Okay, but that's probably not a good idea to be eating and ingesting large sums of that either. So the idea at this point is to look at what is my detox doing? How is my detox working? What do I need to do? And detox from methylation also goes through another set of genes, which would be CBS and CTH. Okay, CBS and CTH take homocysteine and convert it down into cysteine. Cysteine then combines with glutamine and becomes glutathione. Glutathione is one of the main antiviral, anti-detox, detox chemicals in your system that your body's supposed to make on a regular basis. It's controlled by a number of different things and it's found in nearly all the tissues. So it's not just the liver, it's found in the nervous system, it's found in the blood system, it's found in tissue, it's found all over. So when you look at these, you have to understand, you're not dealing with one piece. People talk about detox as if it's some sort of pleasant process. If you're doing detox right, you should feel horrible. You should be sick as a dog because you're dumping toxic chemicals into your bloodstream, into your bowels, your urine for removal. If you feel good detoxing, you're not detoxing.
What about detoxing for gallbladder?
You can't really detox for gallbladder. There's some stuff out there with olive oils and stuff that they use, and you get stones and things like that. Nobody's ever demonstrated if those stones actually come out of the gallbladder. Nobody knows. Sometimes they do, sometimes they don't. A lot of times putting in certain oils and fats together in that combination will create little stones in your bowel, and that's what you're seeing come out the other end. So you don't really know what it's doing unless you're backing it up by looking at the research before and after. Gallbladder scan, then another gallbladder scan. If you see a big difference, then yes. But a lot of these protocols you'll hear out there, a lot of these herbal remedies and deworming and this and detox, they have specific uses for specific conditions, for specific chemistries. If you don't take that into consideration, detoxing alone will not do what you think it's going to do and can hurt you in the long run.
Doc, someone mentioned MTHFR A1298C with uric acid and gout, and wondering if there's a link.
Gout is more related to the fault in purine nucleotide metabolism. When you break down purine nucleotides, ATP and GTP, you tend to produce uric acid, which is one of the ways that the body removes that. There are about 50 genes I can think of off the top of my head that have to do with uric acid. There's an SLC29A, I mean, there's an AP one, there's bunches of these that all relate to uric acid metabolism. Now, does that mean the uric acid is not related to MTHFR? No, that's not necessarily true. If your MTHFR is jammed up, and if you basically turn around and have a problem metabolizing it, then you will not allow two other pathways to go. One of them is called TYMS. TYMS is basically a gene that controls what they call uridine and cytosine metabolism, which are pyrimidines, sister chemicals to purines. Purines and pyrimidines together make up your RNA and DNA. Okay, so if you overload one set or overload another set, you imbalance its removal, and therefore you can get gout from it. It's a bit of a stretch. More likely, the gout is more related to purine metabolism being overloaded because you're having a problem producing enough GTP.
Okay. What about fatigue and high homocysteine levels?
The fatigue and high homocysteine levels. High homocysteine levels mean that the whole methylation cycle is jammed. The methylation cycle, which is not MTHFR, is actually methionine picks up an ATP molecule, the MAT1A gene drops it into basically SAMe. SAMe then methylates and becomes S-adenosyl homocysteine. The S-adenosyl homocysteine becomes homocysteine. That has to do with the AHCY gene. Okay, so there's a bunch of things in there. So the homocysteine, if it keeps jamming up and it can't get out of the system by going through cysteine and the CBS, CTH genes, and it's not dumped out as a sulfite, okay, hydrogen sulfide gas or hydrogen sulfur into the bowel, then basically it cycles back up to methionine. The more you cycle into the methionine, the more ATP it requires to make SAMe. Right. So the more ATP is drained out of your system. You lose ATP, you lose energy. Now you get more tired, your body becomes more tired, your chemical mechanisms slow down, you kind of beat up the system pretty good.
Is that where the body pain comes in?
Body pains can come in from any number of
places. From the drop of the adrenaline?
Well, when you have too much methylation and you break down all your neurotransmitters, you basically lower down the production of neurotransmitters by increasing their breakdown to 10 times faster. As you do that, what you're going to do is basically lower the dopamine, noradrenaline, and adrenaline sequence. Adrenaline is implicated in predominantly pain suppression. If you think of somebody who gets really angry, punches a hole in a wall, breaks their hand, doesn't feel it for 2 hours, and then is, oh my God, I broke my hand. That's basically because while they're pissed off, the adrenaline is firing like crazy. They don't feel anything. Right. That's why dentists, when they give you a shot of lidocaine, there's always epinephrine in it. Why? Because the epinephrine helps deaden the pain to the teeth. The lidocaine is minimally effective. They have to use 3 times the dosage if they're not using the epinephrine. Yes, that pertains to you.
I was just going to say, we'll talk about that later.
When you're looking at how these pathways function, like you don't have an MTHFR. What you have is a CBS CTH double stack. So you can't get out of your methylation cycle. It stays in one cycle over and over and over again, causing SAMe to be produced so excessively that it tears out all your neurotransmitters and basically lowers down hormones. All of these things happen. This is why that's occurring in the system. You don't have to have an MTHFR gene to be a hypermethylator. You could have a CBS-CTH combination. Okay, that means you're just going to keep cycling.
Not getting out of my own way.
Right. And the more methionine you eat,
Right.
the more you keep adding to that cycle, which can't go away. So the bigger that cycle gets, and the more ATP it pulls down. That's where you start running into these excess problems with symptomatology. Excess firing of adrenaline into the system can cause anxiety and temper flashes. Why? When the adrenaline burns out, you're generally in the flight portion of the fight or flight response. When the adrenaline is high, you're in the fight portion of the fight or flight. Now, if the methylation is firing on and off, you're going to keep bouncing back and forth between the two. And you don't know why, but this is also why, if you notice, when your methionine is really high, you get that sudden urge where you really need to have a good fight with somebody. Mm-hmm. You just want to argue and just blow off steam, or you want to go to the gym and work out for 3 hours, okay? You want to dump that excess adrenaline. That's part of the way you can control your methylation defect if you're a hypermethylator. Now, what's a hypomethylator? Hypomethylation is an entirely different type of ball of wax. What it implies is that either A, in its simplest form, the methionine to S-adenosyl methionine doesn't have enough ATP, and therefore it doesn't become active, and you have very low methylation going on in your system.
Right.
So that's one possibility. The other possibility is your homocysteine, your CBS, and CTH are jammed up, which causes basically the methionine cycle to get stuck at homocysteine, in which case now you don't have any methylation chemistry going on and it's not working.
So that's my daughter.
Yeah. Then you have the COMT gene, the PUMT gene, the HNMT gene. These guys all require methylation. If you're not able to make S-adenosyl methionine to be the main methyl donor for all of these, you're going to get symptoms from every single set because it's going to run sluggish. People who drink a lot of tea, for instance, if you have the variant that lowers the COMT activity, drinking lots of tea, okay, inhibits COMT.
Oh.
So that'll make you worse. Okay? Having a low-acting COMT and drinking an adrenaline analog, like your Red Bulls and your caffeine coffees and things like that, means you can't get rid of it as quickly. It's going to stay in the system, and that's going to make you jittery and crazed. So these things all are tied together, and you have to look at the whole system as one unit, functioning as one unit, rather than the system just basically one single thing, one single piece, one single solution. There are no easy solutions here.
Someone asked about anxiety, which is my former life, is a warm feeling in the tummy when they get scared. Could you explain that biochemically?
That warm feeling is one of many feelings. Some people get nauseous, some people get cramps, some people get a warm feeling. All of these occur. Now, why would you have a warm feeling? When you have anxiety, your adrenaline tends to run on the lower end. Okay, it's basically already spiked and now it's crashing. Okay, when it's crashing, the methionine cycle basically has to refill and turn back on to make more adrenaline, which has to come from tyrosine, the amino acid, going to dopa, going to dopamine, going to norepinephrine, and then epinephrine. So that sequence takes a while to get going. So if you burned it all out and you're anxious, you now don't have the ability to break down histamine correctly and balance histamine. Think anaphylactic shock. Person eats a nut, head swells up, throat closes, doctor gives them a shot with the EpiPen, saves their life. The anaphylaxis is a massive release of histamine at one time. Okay. The EpiPen is epinephrine, adrenaline. So when you put the two together, they cancel each other out. Now, physiologically in your body, you're supposed to be doing that all the time. Okay? So what happens if you have no adrenaline? Then your histamine levels are going to be high. What does histamine do? Well, histamine does a lot of things besides give you allergies. Histamine turns on the stomach acid, okay, and that can cause GERD, acid reflux, nausea, and all of that. It also causes vasodilation. Blood vessels get much larger. So you can get a warm feeling in your gut because the histamine level is massively released and the blood vessels are dilating and you're feeling all the blood rush there. That would more than likely cover that.
Okay. And then I have one more question. It was about almond milk and like a head burning feeling.
The
and,
almond milk.
And could it be homocysteine levels?
No. Almond milk really, you're not really dealing with homocysteine at that point. Almond milk has a lot of different things in it. Mostly, almonds are very high in aspartic acid and arginine. Okay, aspartic acid and arginine function in a number of different things. They're part of what is called the urea cycle. Okay, the urea cycle helps pump out nitrogen and ammonia waste from your system and dump it through your urine. But the urea cycle also contains something called the eNOS system, which is nitric oxide. That nitric oxide has a lot to do with how your blood flow goes, where it goes, what it does. So if you suddenly put in almond milk and it kicks up your nitric oxide, you're going to get this feeling of basically blood flow expanding in all of your blood vessels. That will give you a warm, tingly feeling all over. Kind of the similar feeling you would get if you ate turkey. Turkey makes niacin, and niacin can cause what is called a niacin flush, which is why some people will eat turkey and say they're all flushed. It's not an allergic reaction, it's your body making niacin because it needs to get rid of histamine, and it's going to use the niacin to do that.
So wait a sec. So I always run a higher histamine level, but I eat tons of turkey and I never have any type of flush from it.
Right, and you wouldn't because you have high adrenaline. Okay.
I have a lot of adrenaline.
Right. When your adrenaline is firing high, histamine is canceled out. Okay. So that's why you can't look at a histamine test and say, oh, it's within the normal range. Well, it's in the normal range, meaning you don't have a parasite, which eosinophils and histamine levels are related to. You don't have a bug, like Entamoeba histolytica, which produces histamine, which would be medical conditions. Okay. However, if it's in the normal range and it's moderate and you have no adrenaline, then you're going to have allergic symptomatology that no antihistamines seem to help, because it's not a histamine problem, that's normal. It's an adrenaline problem. So this is where you start looking at overlaps on all these different conditions and all these different pieces. They work differently in different parts of the body to do different things. Adrenaline is good because it balances histamine. It's also bad because it causes tachycardia. Lack of adrenaline, you can have massive tachycardia. Lack of balance in adrenaline, okay, where it's bouncing up and down, the tachycardias are going to show up here and there. Does that mean that it's a heart disease at that point? No. That's usually when you go to the cardiologist and you look, and the cardiologist goes through all the tests, and there's nothing wrong with your heart. You get these unknown massive tachycardias every time you get stressed. Well, that's because you're firing adrenaline and it's spiking in the system. That's what's going on with that.
So one question that I get a lot is, why are you different from other doctors? And I always say that you read between the lines of blood tests. So
it's not really reading between the lines. When you look at blood workup, you have to remember that blood workup, and we're going to get into more sciencey stuff here. Blood workup is an average of 1,000 people. There is no standardized piece for blood workup. Blood workup in the United States looks different than the blood workup in Europe, which looks different than the blood workup in South America, which looks different than the blood workup in Asia. Okay, they're all different, slightly different normals because they're based on the population there. So right off the bat, blood workup is not written in stone, which is why they call it the art of practicing medicine, and they don't call it the science of medicine. When you look at things outside of the normal range, you're looking at a medical condition. That's something that a medical doctor needs to look at and understand. He's looking for a bug, he's looking for a disease, he's looking for bacteria, a virus, something along those lines. Okay? When you look at a blood workup regular and you see most of the values are high normal, implying high metabolism rate, you have a few of them that are low normal. More than likely, they are not full metabolisms up to the other part of your metabolism, and they're wrong. And that's the ones you tend to focus on and look at, why is that going on? Once you see that, you can balance that off. Having looked at many, many amino acid scans over the years, literally thousands of them, tens of thousands of them, you get an understanding of what the metabolism is doing and why these values show up. And that's how you can basically see things a little differently. Plus, my background Undergrad is more chemical. My early days with both the Molecular Academy through Huxley Institute kind of give you a different approach and a different look at things. You look at the biochemistry as being part of a problem. Now, is mine the first stop? No, mine should probably be the last stop. After you've had every medical doctor run through all the gambit of tests and look at all the different things and ruled out all the different diseases, at that point, you know it's something metabolic. Medical doctors don't want to deal with metabolic because they've got their hands full with every disease and everything else on the planet, which takes up more time than anybody can possibly imagine. And they turn around and have to deal with the medications and the contraindications and the problems with that, which is another whole ballpark for them to deal with. So as far as metabolism goes, that's not their best thing. That's what makes me more different.
Well, that is part of what I have been listening to since I was 24, and I hope this helped some of you or brought some new questions to light or a little less anxiousness about certain things and some clarity. And that's all for now. So thanks, Dr. Dulin.
It was a pleasure.
Appreciate it.
As always.
All right, everybody. See you soon.
Bye-bye.
Bye.